So I've had some questions about this passage, and to clarify some of your questions, I'm going to go ahead and re-explain some of this. Uh, so this passage is one of the sections in the ACT, and it's called a conflicting viewpoints passage. And a conflicting viewpoints passage is identified by having two different scientists' opinions, or two different students' opinions, or two different experimenters' opinions. It's also characterized by 7 questions, and it's the only one of its type out of the 7 sections. So there's only one conflicting viewpoint. So this is going to be one of the hardest sections there is. The best strategy for doing one of these sections is to read through and try to identify what the important things are, and then kind of summarize as you're going along so you get the idea. So I'm going to read through here, and I'm going to tell you what I think is the most important. So if we read through, it says a polypeptide molecule is a chain of amino acids. So we know that a polypeptide is a chain of amino acids. A protein is one or more polypeptides. So protein is one or more polypeptides. And then we talk about the structure. We have a primary structure is a sequence. And sequence means it's 1 and then 2 and then 3 and then 4 and then 5 and so on and so forth. So it's a nice sequence. And so a secondary structure is folding. So what does it look like when you fold something? When you fold something, it looks like you're going up and down and up and down and up and down. And so this would be like this folded, and those folds are because of hydrogen bonds, weak chemical bonds. So they're hydrogen bonds, so those can be broken. We have a tertiary structure. Tertiary is 3, right? And so we have tertiary structure, which is more folding, right? So this might look like a folded fold. And so we've got a folded fold, and these occur across greater distances than those associated with secondary structure. So greater distances, meaning they're a little bit more complex, and they're folding along those side chains. Quaternary structure, this fourth, is a clustering between more than one folded polypeptide. So this is going to be a folded polypeptide here, and a folded polypeptide here, and another folded polypeptide maybe in front of them. And so it just ends up being this nice big blob kind of protein. And so we have these proteins, and they can have different shapes. Each shape has a relative energy, so energy is what we're talking about in this. And so some shapes, lower energy shapes, are more stable. So less energy, more stable. Less energy, more stable. They are more stable than higher energy shapes, and a protein with relatively high energy shape may denature or unfold. So high energy will break apart. It will unfold and then renature to a more stable shape. So these proteins want to make themselves more stable. A protein that is almost completely denatured is called a random coil. So if it's been unfolded, it would be called a random coil. So unfolded random coil. Random coils are unstable because they're high energy shapes. High energy. And they can renature, adopting more stable shapes. So now let's look and see what Scientist 1 is saying. Scientist 1 says active shape, which is the biologically functional shape, is the identical to the protein's lowest energy. So active shape equals lowest energy. Any other shape would be unstable because a protein's lowest energy shape is determined by its primary structure. Its active shape determined by its primary structure. So determined by primary. So the active is the lowest, and it's determined by the primary structure of that protein. Let's look at Scientist 2's viewpoint. Scientist 2 says the active shape is dependent upon the primary structure. So they're saying the same thing, right? Active shape is dependent on the primary structure. However, a protein's active shape may also depend on its process of synthesis. Process of synthesis, the order in time which the amino acids were bonded together. So, order of amino acids, which is that primary structure, right? The primary structure is the sequence of amino acids. So, the order of the amino acids is going to have something to do with this primary structure. As synthesis occurs, synthesis is creation, right? Making something. As synthesis occurs, stable local structures form within short segments of the polypeptide chain due to hydrogen bonding. These local structures may be different than the local structures associated with the protein's lowest energy shape. So what this scientist is saying is that the primary structure may not be lowest energy. Alright, so he's saying the primary structure is not the lowest energy. It doesn't have to be the lowest energy. These structures persist, trapping the protein in an active shape that has more energy than its lowest shape. So he's saying that the active shape does not have to be, so it doesn't equal, or it does equal, the lowest energy. So those are the two. Scientist 1 says the active is the lowest and it's determined by the primary structure. Scientist 2 says the primary structure does not have to be the lowest energy. It does determine that active shape, but that active shape does not have to be the lowest energy. So let's look at our questions. Question number 1. According to the passage, protein shapes with relatively low energy tend to do what? If they have low energy, we'll go back over here. Low energy shapes are more stable. If they're more stable, they're not going to be random coils. Remember, those are unstable. They don't lack a primary structure. Primary structure is probably the most stable. They don't become denatured because they're low energy, so it's got to be maintain their shape. They're the most stable. Look at number 2. The information in the passage indicates when a protein is completely denatured, it still retains its original what? So when it's completely denatured, it may completely denature and then renature, adopting a more stable shape. If it completely denatures, it's called a random coil. Random coils are unstable. So we adopt a more stable shape after we denatured, and so that denaturing is still going to turn it into a protein. And so if it's unfolding, quaternary structures are folded, tertiary structures are folded, secondary structures are folded. The only one that's not folded is the primary structure. Look at question number 3. Scientist 2's views differ from Scientist 1's views in that only Scientist 2 believes that a protein's active shape is partially determined by its what? Well, we go down to Scientist 2, and he says a protein's active shape may also depend on its process of synthesis. Process of synthesis is not in Scientist 1's opinion, so we can assume that that is the correct answer. Look at number 4. A student has 100 balls. The balls are various colors. Chooses 15 balls and aligns them in a row. So we've got one ball here. We got another ball here, all aligned in a row. If each of those represent an amino acid, what does that structure look like? The spatial order in which the balls were placed corresponds to which of the following levels of structure in a protein? Well, a sequence is going to be the structure, this first structure. If it was folding, if he was wrapping it around the balls, then we'd have another structure, but this is going to be a primary structure. Probably the hardest question on here is this question number 5. Number 5 is talking about these diagrams, and it's asking us which of these is consistent with Scientist 2's assertions about the energy of proteins, but is not consistent with Scientist 1. Okay, so let's first say what Scientist 2 says about energy. Scientist 2 says that we have primary structure is not the lowest energy, and the active shape is not necessarily the lowest energy. It could be, but it doesn't have to be. So he's saying that active shape and lowest energy are not the same. Right? Lowest energy is also the most stable. So let's look at our passage here. Look at our passage. We've got the randomly coiled shape, we've got an active shape, we've got the most stable shape. So right away, we're looking at energy on this graph, energy and which shape it is. And so if we look at the energies, Scientist 2 we know says that the active shape is going to be low energy. And so we know that the energy of the active shape cannot be high, so this is the highest. So we can eliminate A, because that's not Scientist 2. Scientist 2 says that the active shape is going to be a lower energy. All right. He also says that the active shape is not going to be the lowest energy shape, and those two are the same. So the lowest energy shape, most stable shape, is not going to be B, because that's the highest energy in this graph. Let's look at C. In example C, we've got the randomly coiled shape with the highest energy, and that's pretty accurate. But let's look at this active shape and this most stable shape. Scientist A says that the active shape is equal to the most stable, or the lowest energy. So this is more like Scientist A. Which leaves us with D, which says that this active shape is a little bit higher energy than the most stable shape, but it's still the active shape. And that's what Scientist A said. He said the active shape does not equal the lowest energy. And so we can assume that D then is the correct answer.